And before we start, I'd also like to thank Rick Godley. Rick is the founder of the Association of Migraine Disorders, which is Migraine Science Collaborative's parent, and founded MSC, and also really was the one who pushed this contest forward and helped develop it into this very nice sort of annual event that we have. So Rick, thank you as well for all of your efforts.
Thank you, Neil.
Okay, so up first is Yayu Hu. So Yayu, if you can please unmute and make your slides visible, that would be great. Okay, perfect. And if you could put them into, yeah, there we go.
So before I get started, I just want to mention that my voice may sound a little bit scratchy because of a cold. So please bear with me for 6 minutes. Should I start?
Yes, please go ahead.
Okay, so my project is focusing on the role of HPA signaling melanocortin system and basolateral amygdala dendrite changes in repeated stress-induced migraine-like behaviors. We all know that stress is one of the most common triggers for migraine, but we have no idea how stress triggers migraine. We recently published this paper showing that glucocorticoids is involved in stress-induced migraine. We gave metarapone, a glucocorticoid synthesis inhibitor, to inhibit the glucocorticoid synthesis. Therefore, it can prevent migraine attack. So based on this study, we are hypothesized that we already know metarapone can block the mechanical hypersensitivity through the reduction of CORT, but we also know that when CORT reduced, it won't be able to exert the negative feedback inhibition, which can cause the ACTH level and its cleavage product alpha-MSH to increase. So we are curious that the metarapone can block the mechanical hypersensitivity is because of the reduction of CORT, the elevation of ACTH alpha-MSH, or both of them. To study this topic, we use our preclinical migraine animal model. In this model, we gave animals 3-day restraint stress, and this animal will stay in their restrainer 2 hours per day for 3 consecutive days. And this drug will be administered after stress. And we evaluate their pain-like behavior by using the von Frey testing, which is the filament to testing their perioptal area. Animals usually go back to their baseline on day 14, and we gave the nitric oxide donor, sodium nitrate peroxide, to test those animals whether those animals are primed or not. So first, we gave the ACTH to see whether post-stress ACTH injection can have any effect on stress-induced migraine-like behavior. Here's the von Frey testing result. We can focus on the y-axis. If the value is reduced, it means the animal feels pain. If the value is increased, it means the animal feels less pain. So we specifically focus on the red line and the blue line. The red line is stress group and the blue line is stress with ACTH group. You can see treated with ACTH can alleviate stress-induced hypersensitivity during the acute phase and the priming phase. Besides female, we also did this in the male mice, and we can see that the result is similar to the females, that treated with ACTH can attenuate stress-induced hypersensitivity in both male and females. We further investigated alpha-MSH, a cleavage product of ACTH. We found a similar result as ACTH. After treating with alpha-MSH in stressed animals, can alleviate stress-induced hypersensitivity in both females and males. So we already know that ACTH and alpha-MSH, they both work, but where do they work? So we specifically focus on melanocortin receptor, and we focus on melanocortin 4 receptor is because this is the only receptor expressing in the sensory neurons. So here we found a similar result as the previous one. After treating with THIQ, MC4R agonist, can alleviate stress-induced hypersensitivity in both female and males. So besides behavior changes, we also want to know whether stress can also affect the brain morphology changes. We specifically focus on the spine morphology changes. And we know that stress can affect several brain regions, including medial prefrontal cortex, hippocampus, and amygdala. And we decide to focus on the amygdala is because amygdala is involved in pain modulation, and also amygdala can respond to the stress. So we do the same stress model as previous. After 3-day restraint stress, we collect the brain tissue on day 1 post-stress and day 3 post-stress. And then we did the Golgi staining to see the dendritic morphology changes. So here the panel A and B showing the images that from control basolateral of amygdala and 72 hours post-stress basolateral of amygdala. From this imaging, you can see that at 72-hour post-stress, their dendritic spine morphology seems increased as compared to control. Following the panel C, we do the data analysis, and we found that at 72-hour post-stress, the total dendrite length and the dendrite numbers of spines significantly increase as compared to control. So here's the conclusion. For the behavior changes, we know that the THIQ, ACTH, and alpha message can all alleviate stress-induced hypersensitivity in both males and females. In the brain morphology changes, we know that at 72-hour post-stress, it can increase the dendritic spine numbers and the total dendrite length. So here's the implication of my study. First, we know that investigate the HPA axis and melanocortin system in stress can clarify how stress induces migraine. And these findings can support innovative migraine therapies using these existing HPA targeting drugs. And second, we know migraine has comorbidity with so many diseases, including anxiety, depression, and PTSD. They all share the HPA axis changes and elevated inflammation. So if we can use this finding, we might be able to apply to this disease and we can find more overlaps. And finally, I would like to thank to Dr. Greg Duser, my mentor, and all the lab members. Thank you.
Great, thank you, Yayu. Judges, any questions? No questions?
Well, I guess I have a question if no one's going to go. Great work, excellent work. Can you tell me a little bit about your model? Was this, were the animals predisposed to migraine? Was it,
Yeah.
or is this a kind of a non-specific finding?
Yeah. So basically we put animal into our restrainer tube for 2 hours. During these 2 hours, those animal won't be access any food or water. And then after 3-day restraint stress, we start doing behavior testing by using the von Frey testing. And the von Frey testing is we use different filament forces of filament to stimulate their perioptal area. If animal shows any responses, we think that those animals are sensitive to the filament stimulation. And this is our migraine model. And besides this, we also use the Graham score. It's observation to those facial expression of animal. So if the animal, like the shape of their eyes become more small compared to the control animal, we think those animals might have pen-like behavior, or their ears rotate away from their face, all these features.
Okay, great. Thanks again, Yayu. So next up, we're going to have Jhoan Aguilar. Jhoan, if you want to show your slides now, that would be great. He's going to talk to us about perineuronal nets.
Great. Good afternoon, everyone. Let me just put this presentation mode. Okay. Hello, everyone. My name is Jhoan Aguilar. I'm in the Perineuronal lab, and today I will be discussing our investigation and the role of perineural nets in migraine models nociception. So what perineural nets are, they're specialized extracellular matrix components that engulf the soma of the neuron. They're comprised of a variety of macromolecules. And we know that they're modulated by matrix metalloproteinases, and they themselves are modulated by tissue inhibitors of matrix metalloproteinases. The presence of perineural nets is very limited throughout the brain. We also know that they engulf parvalbumin neurons, and in the lab, we visualize them using WFA. Perineural nets influence neuronal health and electrophysiological properties. The interesting thing about perineural nets is that they have been implicated in a variety of migraine comorbidities, such as depression, anxiety, and seizures. And in preclinical models, they have been shown to change. And experimental reversal of those changes have been shown to alleviate the pain-like behaviors that these mice exhibit. So we wanted to know if we could see perineural nets in the ventrolateral periaqueduct and VLPAG, ventrolateral periaqueductal brain. And we found them actually in this brain region called the mesencephalic reticular formation. And further characterization of these neurons showed us that they engulfed parvalbumin neurons. And when we looked at the literature to see more about these parvalbumin neurons, we found this paper from 2022 that characterized their connections and gene expression to show that they project to regions important for nociception and headache, such as the VLPAG, the RVM, the VTA, and the lateral hypothalamus. So we wanted to know if these perineural nets would change in migraine models. So to do so, we administered chronic nitroglycerin, which is a human migraine trigger, every other day for 9 days. And on days 1, 5, and 9, we performed von Frey filament tests on the periorbital region of the mice head. And on the right, we see the mechanical thresholds of this behavior, again showing that a higher number indicating less pain-like behaviors and a lower number indicating more pain-like behaviors. And we see that with chronic administration of nitroglycerin, there is lower mechanical threshold in these mice compared to their vehicle counterparts. So at the end of this paradigm, we collected brain tissue and performed immunohistochemistry assays for perineural nets and parvalbumin in the mesencephalic reticular formation region. Right here are some representative images for perineural nets and parvalbumin, and we quantified these images using Polygon AI to find some interesting things. First of all, we looked at the cellular count for perineural nets and parvalbumin to see no changes in treatment for either male or female mice. Interestingly enough, we looked at the fluorescent intensity of perineural nets and parvalbumin to observe a decrease with treatment in both males and females. So seeing this decrease in intensity in this brain region, we wanted to see if decreasing perineural nets in a non-disease state would induce pronociceptive behaviors. To do so, we specifically injected an enzyme called chondroitinase ABC that dissolves perineural nets in this brain region, the MRF region. So in this experiment, after habituating mice to our behavioral paradigms, I collected baselines for mechanical thresholds, heat sensitivity, and cold sensitivity. After that, I injected the enzyme or our PBS, and after allowing the mice to recover, I tested my behavioral battery from days 5 to 11 for every other day. So showing the results of this experiment here, first looking at the von Frey results, we see that with CHABC treatment, mice that are given this enzyme have lower mechanical thresholds compared to PBS mice. Additionally, looking at the heat sensitivity assay, we observed that CHABC mice have lower latency, suggesting more pronociceptive behaviors. Interestingly enough, when we looked at the cold sensitivity assay, we didn't see any changes with CHABC or PBS treated mice, suggesting that this brain region has a nuanced role in nociception. So to conclude, we found a population of parvalbumin neurons that are engulfed by perineural nets in the midbrain. In a model of migraine, we observed a decrease of perineural nets and parvalbumin fluorescence. And in a different experiment, we showed that dissolution of perineural nets in this brain region resulted in increased mechanical and thermal pronociceptive behaviors. So now some future directions that we have is to assess if pharmacological interventions prevent these decreases in perineural nets in the migraine model, and assess if inhibiting enzymes that dissolve perineural nets prevent pain in this nitroglycerin model. And with that, I'd like to thank my lab, and I will take any questions.
I have a question. I saw in your abstract that you had submitted that you had treated with sumatriptan and found that it prevented the decrease.
Yeah, so we have some preliminary results showing that sumatriptan prevents that decrease in perineural nets. I'm still adding more into that experiment to confirm these results.
So I have a question as well. So a lot of the preliminary data and your background data, that's coming from animals. Is there any evidence of a role of these perineural nets from the human condition itself? So I know you've seen in inflammatory pain and neuropathic pain, but this is in rodents. Is there a link from the patient perspective to these perineural nets being involved in mechanistically in these disorders as well?
To my understanding, I don't think that any investigation in humans has been done in the role of perineural nets in pain. I think it's because perineural nets have just recently been put in the spotlight as they've been recently been studied in the last 15 to 20 years.
So as with regards to that question, when you wrote in your abstract about implications, when you said you'd written that they have been implicated in various comorbid conditions to migraine, depression, anxiety, epilepsy, were these rodent models of those things?
Yes.
Oh, okay.
Okay, great. Any other questions? No? Okay, thank you very much, Jhoan. So next up, we have Mainvire Kaur. Please get your slides up. It's going to be looking at some sex-specific responses to cortical spreading depression.
You guys see my slides?
We can see your desktop. There we go.
Okay.
Yep.
Hi, everyone. This is Mainvire Kaur, and I'll be talking about photophobia and pain associated with migraine with aura. To give you a brief background, cortical spreading depression is a neuronal wave of depolarization followed by a quiescence or decrease in cellular activity. This is a mechanism thought behind for pain and photophobia experienced by migraine with aura. What it causes activation of trigeminal ganglia, which sensitize the nerve, and therefore there's a pain in periorbital region, and then there is photophobia. In animals, we call it periorbital allodynia, which is heightened response to a non-painful stimuli, and we measure in this area. Photophobia is the most bothersome symptom experienced by migraine with aura. However, the exact mechanism of CST involved with photophobia is not fully elucidated. So in this study, we hypothesized that male and female responses are different to pain and photophobia induced by CST. We measured this with 3 sets of aims. We looked for trigeminal sensitization, which is just measuring periorbital allodynia. We looked for photophobia, in which we studied development, incidence, and magnitude of photophobia. And finally, we correlated these findings by measuring pupil to iris ratio and level of eye squinting. How we did this? So we did 3 different types of experiments. Before every experiment, we did baseline assessment. This over here on the left-hand side, I'm showing you a 3-chambered box, which has light, transition, and dark chamber. It is equipped with infrared lights, which once the animal enters, it starts measuring the locomotion, which could be inferred into photophobia. So we induce CST by doing a surgery. This apparatus is called a stereotax. So we cannulate these rats on the occipital cortex. And after giving them a 1 week for recovery, we induce CST by injecting KCl, potassium chloride, which causes ionic disruption. And then on the day of experiment, we measure periorbital bone fry, we measure physiological measures, pupil to iris ratio, and finally photophobic assessment. I'm showing you periorbital allodynia graphs on this slide. On the left-hand side, I have % of animals with periorbital allodynia over time. You could see at baseline, females are more sensitive without anything. But over time, after CST induction, you see that at 3 hours to 4 hours, 100% of females have periorbital allodynia compared to less than 30% of males. On the right-hand side, I'm showing a facial withdrawal threshold graph, which is, we measure periorbital allodynia with bone fry filaments, which is specific force, starting from 0 to 8 grams. Any animal which has a degree which responds in this area, 6 to 8, are considered non-sensitive. And we only include these animals after they pass baseline. So you could see after at the baseline, everyone starts at 8, but with increasing time, females are highly sensitive compared to male. Next slide, I'm showing you photophobia. On top is light chamber assessment. On bottom is the dark chamber assessment. In this, we had 3 groups per sex. We had non-injected, but cannulated rats. We had rats injected with ACSF, artificial cerebrospinal fluid, and we had rats injected with KCL. If you look at the males, this is an area under the curve for total time spent in the light box. If you look at the males, there is no statistically significant difference. But if you look at the female graphs, there was a trend. KCL females spend less time than ACSF compared to non-injected, and it is highly significant. Then we use this time and we translate into an index called as aversion index. This index helps us translate their locomotion or photophobia over time. We did this study for 24 hours. For males, you could see there was no significance. But for females started light sensitivity early within 15 minutes, and it intensified over 3 hours and comes down after that. On the bottom is the dark chamber. Similarly, 3 groups, males and females. There's nothing going on with males, but with females, KCL females who had photophobia and more pain, they spend more time in the dark chamber. And then we use a dark seeking index, again, translated index from the total time spent. Males, they have something at 15 minutes, but nothing else is going on. But if you compare females, KCL females to ACSF and then to non-injected females, they're highly significant. They have increased pain and they're spending more time in the dark chamber. And they recover after 4 hours. But this was done over 24-hour time period. Finally, I'm showing you the graphs from physiological measures. On top, I have pupil to iris diameter. In the naive, male and female, there's no difference. But when we look at males, this is only done for ACSF and KCL. We see at 60 minutes, irrespective of ACSF or KCL, both have decreased pupil to iris ratio. Same things are seen with females. But at 180 minutes, nothing is going on. They recover. When we look at palpebral measure, degree of eye closing, which is also known as squinting, nothing happens with male. There's some baseline differences. But if you look at female graphs at 180 minutes, there is a difference between ACSF and KCL, which also correlates with the highest degree of pain. At 180 degree, females have more eye closure, more photophobia, and high pain sensitivity to trigeminal nerve sensitization. Finally, I'm summarizing and giving my key message that female rats show heightened photophobia and pain sensitivity compared to males. Females had stronger physiological responses and sustained periorbital allodynia in a post-KCL injection CSD model. This graph recapitulates what I said. We start at baseline. At 60 minutes, females have light sensitivity. They also have decrease in pupil to iris ratio, which is in both groups, males and females, both conditions, ACSF and KCL. 120 minutes, there is dark sensitivity. They're spending more time in the dark chamber. And periorbital allodynia, 100% of females have periorbital allodynia compared to less than 30% of male. At 180, everything is intensified, and females also have eye closure. Finally, to state some implications of my study, sex is not considered as a common consideration for pain medicine. So precision medicine based on pain mechanism, which are different in male and females, could be beneficial. Another thing like clinical trials, sex male to female ratio should be critical. Finally, some failed clinical trials should be reevaluated with subgroup analysis for sex. With this, I would like to conclude my slides and say thank you. And if you have any questions.
I have a question. You probably had this up there, but did you measure if the threshold in females was lower for CSD than in the males?
We measured, yes. So we measured by periorbital allodynia, and we also measured photophobia, between males and females.
Right. But did you actually look at the tracings of like CSD?
So this method has been validated by our lab. Like we have actually done this before, but not for the current study.
Okay.
But this is validated. We just followed the same regular method. It is published.
I had a quick question. Manvita, thanks for the presentation.
Thank you.
I was just wondering about are there any anatomical differences between male and female rats in terms of, the eye, the light perception, the size of the pupil, presumably the male rats are are you in rats? Yes.
Yes.
Presumably the male rats are bigger.
Yes.
But can you tell us a bit more about that?
Yes, you're right. Male rats are way bigger. They're like start at 250 to 300 grams than females.
Right.
Everything is big in them as compared to females. And there is a difference. So before doing any of the photophobia experiment with surgical cannulated rats, we did just with males and females. And it was interesting that males spent more time in dark. And females, they were okay because nothing was done to them. So natural preferences are different between males and female rats. Anatomically, yes, they have more space because they are bigger. And then neurologically, we know there's so many, like the pain receptors for male and females are different. Like there was total differences between males and females.
So then I suppose, how do you think about those anatomical differences versus, something else that would be intrinsic to responsiveness to light stimulation?
So when they did not translate it into any pain or any photophobia, irrespective of those differences. Even they were bigger, they were given similar amount of KCL, ACSF, similar amount of time. So this did not lead to any changes in post-experimental study, but same animals had differences before actual surgeries. So I don't think there's any anatomical differences makes any difference. I feel there was enough more to it, like more hormonal differences that could play a role in pain and photophobia.
Okay, thank you very much. Mm-hmm.
You're muted. I'm gonna stop
Sorry. Thanks to the migraine group for the 3 presenters. That's great. So now we're going to move on to our next group, the pain group. We have 3 more presentations. So first up is going to be Saurav Gupta, who is from UAB and is going to tell us about trigeminal ganglion.
Are you able to see my screen?
Yes.
Okay. So is it good, right? My sound is also good.
Yep, perfect.
Okay, perfect. So my title is like characterization of electrophysiological property of trigeminal ganglion neuron that innervate the maxillary sinus in mice. So for the introduction, this maxillary sinus. So this is one of these like paranasal sinuses, which is like innervated by this trigeminal nerve. And this maxillary sinus is located in like this location where you can see here is the like a airfill space kind of one. So this function usually does the lightening skull weight, moisturizing and cleaning the nasal cavities, and suppresses this infection by producing mucus. And these all are like innervated by this, like most of these maxillary sinus are innervated by the infraorbital nerve. So this is the nerve which is the B2 branch of the maxillary nerve. So for this like a maxillary nerve, they are like involved in causing disorder like sinusitis, migraine, nasal congestion, and rhinorrhea. And so it generates which causes by generating noxious expression and persistent pain. And for migraine, it can disrupt the normal functioning of the paranasal nervous system, leading to symptoms that closely resemble those of sinus infections. And this chronic pain generated from these maxillary sinuses is often poorly treated, partially because of the lack of the knowledge on the trigeminal nerve that convey nociceptive signal from the maxillary sinus. So because of that, we aim to bridge this knowledge gap by characterizing this maxillary sinus innervating neuron and to identify the gene expression profile associated with mechanosensitivity and nociception. So for that, we have this animal model which we're using the wild type mice and the transgenic mice. So for the transgenic mice, we have this SC and ethane acri, which is a voltage gated sodium channel 1.8, and it is crossed to the channel rhodopsin AI32. When you cross this, then we will be leveling these nociceptive afferent neurons, which will help for the precise visualization and manipulation. So we are using these transgenic mice to visualize the nociceptive neurons. So to actually target the maxillary sinus, so we are using this DID, retrograde tracing dye. So DID, it is a fluorescence that labels these neuronal cell bodies from their axon terminals. So here you can see it is a blue dye. So this is the eye socket. Just below the eye socket there's a maxillary sinus, which you can see over this figure, after the injection. And for the recording, we're doing this patch clamp recording. So for the patch clamp recording, we're making this ex vivo preparation, and this is the neuronal cells for the 3G, and this is the infraorbital knob. So we have this elongated infraorbital knob, so we can measure the conduction velocity to specify what kind of fibers is it. So here you can see a bright view of the neuron, and here will be the nav 1.8 a positive YFP this is a visualization of this nav 1.8 positive neurons over here. So for that, now we have this now DID a sinus injection. This is the sinus tissue neuron which is showing in a DID. So if you see the same location over here which is showing negative, and this is a nav 1.8 positive neurons, and what we see that it is showing negative of this nav 1.8. So while you see that most of them are not showing nav 1.8 positively visually in this case. And now we are measuring this conduction velocity for this sinus tissue neuron and this nav 1.8 positive C afferent neuron, and we see they fall under 1 meter per second, showing they are C-type afferent neurons, but the conduction velocity of this sinus tissue neuron is significantly higher than this nav 1.8 C afferent neurons. So this is now showing a distinct characteristics from the sinus tissue neuron, with the C afferent neuron. Now, furthermore, so we are saying okay, these neurons exhibited multiple action potential firing, so supporting their nociceptive function. So for here, we can see this sinus tissue neuron is giving similarly multiple action potential firing, in case of C afferent neurons, which is nav 1.8 positive, and we do see a significantly higher multiple action potential firing in the sinus tissue neuron. And you can see over here, there's no any change in the number of action potential at 2-time reobvious. So there's a single firing. Here is most of them are showing single firing, and 4 of them are showing single firing in this a nav 1.8 C afferent neuron. So we see they have also nociceptive function. So furthermore, we want to see their passive membrane properties, and they did so different passive membrane properties from this nav 1.8 C afferent neuron, where the soma diameter is significantly higher than this a nav 1.8 C afferent neuron, and the resting membrane potential is less negative, membrane capacitance is significantly higher, and membrane resistance is significantly lower. So this is showing sinus tissue neuron is very different from showing different characteristics from this nav 1.8 positive C afferent neuron. And furthermore, we characterize this active membrane properties, and in active membrane properties we see this reobvious is giving the current injection 10, 20, 30, 40 kind of a current. So we did not see any kind of significance showing similarity with this C afferent neuron. Even the threshold is also not significant. Action potential amplitude is also not significant, which is measuring from the resting membrane potential to the AP of stroke. But we see a significantly higher AP width than this than the nav 1.8 C afferent neuron. So from here we can see that this is also playing role like in a nociceptive kind of a feature, and AHP is also significantly more negative than this like NaV1.8, like a afferent neuron. So we did see like some of the properties are showing different from this like NaV1.8 C afferent neuron. And while looking on the Bordes-Cramp configuration, so this synesthesia neuron is really showing that the like significantly higher outward current at the initial phase and late phase, and the higher inward current, where you can see in this figure, and here is the IV plot showing they are significantly, synesthesia neuron is significantly higher in their outward current and higher in their inward current. So furthermore, now we isolated these like a single neurons, like then after isolating these single neurons, so we were able to run PCR to see whether they are like piezo2 positive or no. So we did see this piezo2 gene expression identified in this individually isolated synesthesia neuron using the mRNA and PCR analysis. So this is like 7 sets are done with 7 different from 7 different mice. So we did see like 4 of them really expressing the piezo gene, but 3 of them did not express the piezo gene. So in that way, for the conclusion, we can see that these studies characterize the electrophysiological properties of trigeminal ganglionic neuron innervating the maxillary sinus, and the synesthesia neuron exhibited nociceptive feature which such as like multiple action potential firing, large action potential with then this like NaV1.8 C afferent neuron, and the presence of piezo2 gene expression in this neuron indicate mechanosensitivity, and this finding suggests that the synesthesia neuron are involved in sensory processing related to sinus pain and mechanotransduction, and this mechanosensitivity and nociceptive properties of this neuron may be particularly relevant to understanding how sinus-related pain could overlap with or contribute to migraine pain. So for my acknowledgement, I really like to thank Associative of Migraine Disorder to fund this project and my PI, Dr. Jhoan. So thank you for your attention. I'm happy to take any questions.
Any questions from the judges?
So where do you take this next, having sort of potentially identified these sinus neurons which might be implicated? Where would you take this to try and, I mean, I guess it's not necessarily just migraine, it could be implicated in other facial pain or other facial related disorders, but where would you go next with this specifically?
Oh, so for my future direction, I do have plan to like identify this neuron. So I'd like to identify the gene expression profile associated with mechanosensitivity and nociception by individually isolating synesthesia neuron, and I'll also study the presence by using electrophysiologically to see the presence of ionic current mediated by mechanoreceptor TRPA1, TRPV1, ASIC3, and there's also other genes which will be looking for whether they are comes under the TRPA1 nociceptor or a CLTMRs or other things. So we have more further studies to go on for this part.
So this is great. Did you do the electrophysiology yourself?
Yes, this is all done by myself.
Wow, that's a lot of work.
Yeah, thank you.
So I have a couple of quick questions, if that's okay. So one is a little bit technical. Just you mentioned towards the end that you saw a big occurrence in the retrolabeled neurons. Wouldn't you expect that if the cell size is bigger?
Yeah, that was also expected. Since what we find interesting is
and what if you did current density instead of absolute amplitude of current?
Current density is also an option, but we have not tried in our lab relating to that. We do measure the conduction velocity to really categorize.
No, but you can do it because you have capacitance, so you can just normalize by capacitance.
Yes, so it's one of the approaches which my lab has not really used on the current density.
Okay, and then the second question is really related to the number of cells that you're seeing in the trigeminal ganglia that are labeled. What were you expecting to see? Just in terms of the total number that are labeled in that ganglia. Did you have some sense of how many cells would be labeled?
So for that one, it is really like not that much a lot. I usually find like 10 to 20 neurons while on my recording, but it is the preparation is ex vivo, so I can only see the top layer. So I don't really see what's like in the down layer on that part. But while isolating like individually, I was able to isolate around like around 12 neurons on that like
But
while.
it looks like quite a lot. It's labeled. Oh, this is what
No, this is a
YFP.
NAV
So this
1.8
is
positive.
so that's 1.8. Oh, okay. So of the NAV 1.8 positive, so of relative to NAV 1.8, what so you see about 10 to 12 neurons labeled, is that right?
Yeah, it around like
Okay, okay, great.
around
Sorry, I
20.
got confused. Okay, thank you. Yeah, that makes a lot of sense. Okay, great. Thank you.
Thank you, really appreciate it.
Mhm.
Okay, great. Thank you, Saurav. So second presenter in our pain group will be Claudia Daniela Montes Angeles. Claudia is also going to talk about a related topic, orofacial pain. So Claudia, please show your slides when you can.
All right, so please let me know if you can see them.
Yep, we can see them.
All right, well, hello, thank you very much. I am Daniela Montes from Perez Lab, and the title of my work is A Novel Model of Chronic Orofacial Pain for Assessing Pain Phenotypes and its Neural Substrates. It is well known that chronic neuropathic orofacial pain can be produced by an injury on the somatosensory trigeminal system, and since it decreases considerably the quality of life of whom suffer from it, it's very necessary to work in experimental models that resemble as accurately as possible orofacial pain clinical features. And in here, we are proposing the mental nerve compression injury as the model that resembles such conditions. This way, we've been hypothesizing that the mental nerve compression injury is a useful model for studying different phenomena that occur in the development of chronic orofacial pain. As I was telling you, the orofacial pain model was a mental nerve compression injury in which we made two compressions with superfine, highly fine forceps on the mental nerve, which is located right at the exit of the so-called foramen of the mandible, and we characterized this injury both mechanically and histologically. Mechanically, with a pressure sensor, just to make sure that the pressure applied to every single one of our compressions was not different between each other, and the histological characterization was performed to see what would happen with the injured nerve after 14 weeks of assessment. And here we can see that this is representative for all of our mice, in which after 14 weeks, our crushed nerve would remain injured until the end of our experiments. In order to characterize the sensory dimension of pain or of this model, the sensory effects of this model, we performed bone freight test on the mental region that includes the lower lip, the chin, and the lower lip commissure. In here, we assess them for a baseline and from day 3 to week 14 after compressing this nerve. In here, we found that compared to our control group, our injured mice decreased considerably and significantly their nociceptive threshold, and it will remain after several weeks. The very next step was investigating what this model was useful for. And we found that this model is useful for identifying orofacial pain phenotypes and for investigating their neural correlates. We found through bone freight test that all of our injured mice could be classified on week 2, depending on their nociceptive threshold, whether it was high or it was low, and whether or not they would recover their baseline levels. This way, we found that our high threshold mice would recover along the weeks, around week 3 to 4, as we can show in here, and we found that our low threshold mice would remain hypersensitive until the end of our experiments, and we found that most of our mice were high threshold and would recover. For evaluating the neural correlates involved, we used a specific neural ablation strategy in which we injected a viral vector in the nucleus accumbens of the D2 receptor expressing transgenic mice. The effect of this was eliminating specifically the nucleus accumbens D2 receptor neuron expressing D2 receptor expressing neurons, eliminating such neurons. And here we found that eliminating this population in the nucleus accumbens modifies the patterns of chronic orofacial pain phenotypes, making mice more susceptible to chronic orofacial pain and delaying their recovery when the case. The second phenomenon we found that this model is useful for is for evaluating the pain-like facial expression and investigating their neural correlates. In here, on head-restrained mice, we video-recorded their facial expression under the stimulation of bone fray filaments. We video-recorded them, we extracted their frames, and we analyzed them under a machine vision strategy, and we found that the similarity to the pain-like facial expression from a different part of the body that we used as prototype is high, and that the changes, this similarity and the changes from the baseline levels are depending or increase when we also increase the intensity of our stimuli. For evaluating their possible neural correlates, we use a strategy of decreasing the activity of the anterior cingulate cortex by two strategies. The first one was the specific elimination of the ACC glutamatergic neurons with the strategy that I previously described to you. And the second one was with the optogenetic silencing of ACC glutamatergic neurons by activating the GABAergic neurons of such region. And in here, we found that decreasing the ACC glutamatergic neurons activity, the pain-like facial expression occurrence is prevented at least during early stages of or in the onset of chronic pain by day 4. This way, we conclude that mental nerve compression is an accessible and effective model for studying trigeminal pain different phenomena from behavior to neural substrates, and this might be useful for a study of not only neuropathic pain, but also for different craniofacial pain conditions, and this would help us to improve the treatment, predict it, and if possible, maybe prevent it. So this way, I'd like to thank you, and if you have any questions, I will be happy to take them.
I'm going to ask a question. So the original normal orofacial pain models of neuropathic pain normally do, say, for instance, infraorbital nerve constriction. I'm just asking, is this method better? Is this method an add-on? How does this improve, or is it what is it going to tell us that is different to what perhaps the traditional method is using? I mean, normally you would measure whisker pad for infraorbital. Obviously, this is slightly different nerve body.
Okay, well, the first thing that is different from infraorbitary nerve is that it's less aggressive than it, and yet it's still functional for assessing the phenomenon. And this is more, this is a useful model for adapting the neuropathic injuries that are performed, for example, in the dental office for just such as procedures as surgeries, implant collocations, maybe even an improper anesthetical technique. And this is useful also because we found that their outcomes, it's not always that the 100% of these of our mice develop pain in a sustained way. So that's what also happens with patients. Not all of them would develop chronic pain, but also may develop sensory alterations. So it might be resembling in a better way what happens at the clinic level.
Okay, cool.
Any other questions?
I do have another question, actually. So I know you did this imaging of the face to presume to look at sort of facial expressions as an example of pain. Were you able to translate the animals which were high threshold or low threshold to their different expressions? Did they sort of group separately, or had you sort of separated them at this point?
Actually, that's part of the next step of our study, because the first step was identifying whether or not they could make a pain-like phase derived from the mental nerve compression injury. So it's going to be like very interesting to find out if that happens, and if we can use them as landmarks of whether or not they're going to develop chronic pain.
Okay, thank you.
Thank you.
Okay, great. If there are no other questions, we'll move on to our third presenter in the pain group, which will be Jhoan, who's going to be telling us about a human study. Please go ahead.
Oh, okay. Can you see my screen?
Yes.
Okay, thank you. All right. Hello, everyone. I'm Jhoan from UCSF. Today, I will introduce my work in the anterior insular-based network biomarkers of experimental pain in chronic pain patients. So as we know that one important characteristics of the brain's responses to pain is the complexity due to the involvement of nociceptions, different sensations, and even like some motor activities. So when we are analyzing the brain's responses to pain, it is very important to involve different brain regions and different frequency bands. In such a context, I aim to use functional connectivity to measure the integration among different brain regions so that we could use brain network as a signature to see if it can predict pain. According to some previous work, it was suggested that anterior insula has a key role in the pain processing. However, if we use some non-invasive technology such as EEG, it cannot record the signals from insula directly. But benefiting from stereo EEG, we could record neural signals from anterior insula. So that I aim to build the anterior insula-based brain network as the features to input into a machine learning model to predict the subjective pain intensity. Here we use numerical rating scale as the scale. In our protocol, since this is a preliminary study, I used one patient from our clinical trials who was a male at 50 years old with CRPS at the moment when we were conducting the experiment. And we implanted the stereo EEG into his brain. So in this work, we analyzed the effects of experimental pain in such chronic pain patients. We used the thermal to induce thermal stimulation to the patient to both left or right hand. In this protocol, it's called ramp and hold, which contained 4 different phases, including resting state. The onset, we called it ramp, which was always at a constant speed to increase the temperature to a target temperature. The target temperature could vary in different trials. And in each trial, we had a hold session where the temperature remained at the target temperature for 4 seconds. And in the last second, we asked the patient to rate a pain with NRS. And the last phase was the offset. Such trials were repeated for several times following a fixed order in the whole session. After recording the neural signals with SEEG, I preprocessed the data and filtered them into 5 different frequency bands, including theta, alpha, beta, low gamma, and high gamma bands. With these signals, after segmenting them, I measured the functional connectivity between the anterior insula and the other brain regions as a spectral coherence. Here is an example. You can see that it covered like all the brain regions, which was showed here. And the left side represents the left anterior insula, and this is right anterior insula. Each element represents connectivity between the anterior insula and the other contact at another brain region. For reducing the dimensionality of the features inputting to the machine learning model, I used principal component analysis, and then it inputs such an X-ray boost regressor to predict the NRS. By the way, I used leave one out to all the whole trials. All the data are from the whole trials. Because we don't know the NRS of these regions, so we only focused on the whole trial in this research. so that it will beat the overfitting. So here we can see that the performance of the machine learning model was pretty good. It means that such anterior insular-based brain networks can be a good signature to distinguish different subjective pain intensity. And according to the important features exposed by the PCA and XGBoost, I found that the functional connectivity between the anterior insula and you can see the somatosensory cortex was very important. And the theta, alpha, high gamma bands had main contributions in the pain prediction. And now we have another question that since we only know the NRS at the hot trials, can we use such a model to predict the change of the pain intensity? The answer is yes, because I analyzed the correlations between the temperature and NRS, which had positive correlations. So it means that the change of the pain intensity in such ramp and even offset is feasibly to be predicted with such a model based on the anterior insula-based brain network. So in the future, following this work, I will use our data from the experimental pain induced with the thermal from different protocols and the mechanical sensations effects with one-free tests. In the future, based on this research, it will be possible to disclose neurodynamics in the change of pain. Thank you all, and especially thanks to my supervisor, Dr. Prasad Shivakar. Thank you.
Any questions from the judges? No questions. Okay. All right. So
I have a quick question. I'm sorry.
Oh, sorry.
I was a bit
Diane.
slow.
Yeah.
Just to, so I'm interested in the application of this in to non-invasive type of assessment of neurodynamics. So, are you thinking that ultimately you should be able to measure some kind of, or get some signature of the neural network, and then be able to make a prediction about how much someone is in pain, or is it the reverse order? So right now you're doing it, you're measuring this activity through implanted electrodes, right? So
yeah.
What kind of leap would you make in order to be able to do this in a non-invasive way as a diagnostic?
Okay, so first, because our clinical trials is for the neuromodulation with DBS, deep brain stimulation. So these are just in the pre-surgery. We just made this test to collect the data, to show the neuromechanisms, and to find which signs are important for their further implantation for neuromodulation. And as to your question, because here I think so first, this data can cover the, enough brain regions, and it can build, so because this is very preliminary, now I'm extending it to the whole brain regions, the connectivity between among all the regions. So in the future, since we could detect the change during the onset of pain, we could even in the future, it will be feasible to detect the real onset of pain, when they really perceive the pain. So I think this is, how to say, although we don't have labels about where it came, but I could in a data-driven way, I could expose different brain network patterns related to different parts, like nociception, thermoception, and the raise of pain. I don't know if it can explain the answers. Sorry.
Yeah, I guess so I understand the methodology in the DBS for doing neuromodulation, but I was wondering whether there's a way for you to leap into a more non-invasive kind of assessment of pain thresholds and intensity of pain. That was the question. It sounds like you're not there yet. That wasn't the intention of the study, but there is a possibility
Yeah, of course. I think that's generalizable to the non-invasive technology because I worked with EEG before. I know that although their coverage to the brain was different. So for SE, kind of like cover the occipital lobe, not cannot. It was not necessary. But in this, I think the main purpose is to find like which kinds of networks are responsible for which parts of the pain processing.
Sure.
So under such a context, I think I could use that to use the data from, the non-invasive technology, even from like healthy controls, from like, the experimental pain induced to general healthy populations to check if that's really generalizable. It's not like some personalized thing. Yeah.
Thank you.
Okay, thank you very much. Thank you.
Okay, great. If there are no more questions, let's move on to our final three speakers who are in our pharmacology group. So first up will be Yoshihiro Kitaoka, who is from UCLA, is going to talk about cannabinoids. Please go ahead.
Yeah, can you see my slides?
Yes. If you can put them in slideshow mode.
Yeah.
If you can.
Yes.
Yeah.
Yeah. Okay, so yeah.
Perfect.
Okay, so good afternoon. So I am Yoshihiro Kitaoka, a postdoctoral fellow at the Laboratory of Neuropharmacology, UCLA School of Dentistry. Thank you for giving me the wonderful opportunity to present. Today's presentation title is Peripherally Acting Cannabinoid Prevent Migraine-like Pain and Sensitization of Dural Nociceptors. Yeah, let's get started. Migraine is a debilitating neurological disorder that affects up to 50% of the population globally, with greater prevalence in females. Cannabinoids acting on GIO-coupled cannabinoid 1 and 2 receptors alleviate migraine symptoms in human and in animal models. However, side effects mediated by CB1 receptor in the central nervous system limits their widespread use. We developed a synthetic peripheral restricted cannabinoid, PRCB agonist, that do not cross the blood-brain barrier. The lead PRCB, PRMNI, effectively suppress the chronic pain symptoms in preclinical model of cancer, chemotherapy, and traumatic nerve injury-induced neuropathies, all with minimum CNS-mediated side effects to tolerance development. We use a non-invasive mouse model of dural stimulation to induce migraine-like pain symptom of periorbital alveolar and latent sensitization to mild stimuli. This model allowed us to address the mechanism of trigeminal dural afferent sensitization and the utility of PRMNI as a pre- and post-treatment in suppressing behavior symptoms of this sensitization. We used 5 weeks old female C57 Black 6 J mice for all experiments. We performed supradural injections through the cranial suture at the bregma using a 33-gauge cannula. We injected approximately 4 microliters of saline at either pH 6.0 or 7.4. A second injection with pH 7.0 saline was administered 72 hours later. We measured head withdrawal threshold in response to periorbital stimulation with calibrated foam plate filaments to evaluate the mechanical sensitivity. Behavior tests were made by investigators unaware of the type of treatment given to mice.
Yoshihiro, I'm sorry to interrupt.
Yes.
I've lost your slides on my screen. Can people see the slides or no?
Yes.
You can see them? Okay, must just be my screen. Okay, go ahead. Sorry for the interruption.
Okay, no problem. So pH 6.0 solution caused pain, and the pH 7.0 solution, mild stimuli, caused latent sensitization. We also performed wholesale patch-clamp recording on flow gold-positive trigeminal ganglion neurons to study the mechanisms of latent sensitization. We use the PRMNI, the CB1 receptor antagonist 18A, and the CB2 receptor antagonist SR144528. These drugs were dissolved in pH 6.0 saline for superdural injections. Co-treatment with PRMNI prevents acid-induced hyperalgesia and latent sensitization by CB1 and CB2 receptor activation. Selective CB1 and CB2 receptor blockers prevent the analgesic effect of PRMNI. These data suggest that PRMNI effect are mediated by activation of both CB1 and CB2 receptor in this model. However, post-treatment with PRMNI did not prevent the latent hyperalgesic response to a mild pH 7.0 stimulus. Majority of flow gold-positive Tietjen neurons were labeled with isolectin B4, which labels a subpopulation of pain-sensing neurons. All flow gold-positive neurons exhibited characteristics of A delta type and C type nociceptors. The A delta type can be mainly distinguished from C type by presence of a voltage sag in response to a hyperpolarizing current pulse. I applied the pH 6.0 solution to the bath 3 minutes and investigated the acid response of Tietjen neurons from pH 6.0 and pH 7.4 treated mice. Tietjen neurons from pH 6.0 treated mice exhibit larger depolarizing response to pH 6.0 applications than neurons from pH 7.4 treated mice. This suggests that increased functional activation of acid-sensing ion channel contributes to the behavioral symptom of latent sensitization. We also began investigating a PRMNI effect on trigeminal neurons and showed that PRMNI reversibly decreased response to pH 6.0 of flow gold-positive A delta type Tietjen neuron from a mouse that received a superdural pH 7.4 injection. These experiments are ongoing. In conclusion, acid-sensitized dural nociceptors are a major contributor to the migraine-like pain triggered by mild stimuli, which can be prevented by pre-treatment with a peripherally restricted cannabinoid receptor agonist. This research can potentially improve treatment option and enhance the quality of life of patients with migraines and related conditions. Thank you for your attention.
Thank you. Any questions from the judges?
I can ask a question. So this is the very first step, I guess, because you've given, if I'm not mistaken, local injection of the pH 6, and then you've also given local injection of the cannabinoid. What is going to be your next step? I mean, you're saying this is a potential treatment, but you're sort of over here when, you know, treatment's over here. What is your next step of what you're going to do to try and support what your future perspective is?
Right now, I'm doing also electrophysiological recordings, so patch clamp recordings. So how PRMNI affect the acid-sensing ion channel? So based on the pH 7.4 and pH 6.0. So let me show the last slide.
Okay.
So
thank you.
So this is for the only one data. So this experiment is ongoing. So we need to get more data for this, how PRMNI affect or not.
Okay, thank you.
All right. If there are no other questions, let's move on to our second speaker in this group of pharmacology. So next up is Yoonjin Go, who will be talking about GLP-1 in the news a lot. Please go ahead.
Hello, everyone. Can you guys hear me?
Yes.
Okay. Hello, everyone. My name is Yoonjin Go from Gachon University, College of Medicine in South Korea. Today, I'm going to talk about how GLP-1 and its derivative peptides contribute to pain relief through the TRPV1 channel. Have you ever noticed that eating high-calorie or high-fat foods makes you feel better when you get cold and in pain? This phenomenon known as ingestion analgesia has been observed in studies where mice showed reduced heat sensitivity after consuming chocolate or sucrose. While most research has focused on how the central nervous system mediates ingestion analgesia, we wanted to explore whether hormones secreted during digestion might play a role in suppressing pain through the peripheral nervous system. Among the many reactions during digestion, we focused on GLP-1. This hormone is secreted in the gut, stimulates insulin secretion, and lowers blood glucose levels. GLP-1 analogs were initially developed for diabetic treatment due to its short half-lives. Recently, semaglutide, which is another GLP-1 analog, gained attention for its use in weight management beyond diabetic therapy. In our study, we observed that intraperitoneal glucose administration reduced heat sensitivity in mice as measured by hot plate test. Similarly, GLP-1 administered intraperitoneally, orally, or directly into the paw showed consistent pain relief. Since heat sensitivity is linked to the TRPV1 channel, we tested the effects of GLP-1 with capsaicin, which is TRPV1 activator. And mice were co-injected with GLP-1 and capsaicin exhibit significantly fewer pain responses compared to capsaicin alone. These results suggest GLP-1 modulates pain through TRPV1 interaction, highlighting its potential for pain relief. To understand the mechanism, we cultured primary DRG neurons and analyzed their responses to capsaicin using patch clamp and calcium imaging. We found that GLP-1 and its agonist, such as exendin-4 and liraglutide, inhibited TRPV1 activation induced by capsaicin. Since GLP-1 receptor agonist inhibited TRPV1, we hypothesized that the GLP-1 receptor mediated this effect. However, as you can see, when we used the GLP-1 receptor antagonist, exendin-939, it showed even stronger TRPV1 inhibition and reduced pain responses. This suggests that exendin-939 works independently of GLP-1 receptor. Next, we focused on exendin-939 and tested its effects in inflammatory and neuropathic pain models. In both models, it significantly reduced pain behaviors such as paw withdrawal latency and threshold, demonstrating its strong analgesic effect. And unlike other TRPV1 inhibitors, exendin-939 did not cause hypothermia, and it selectively inhibited TRPV1 activation without affecting proton-induced activation mode. These results show that exendin-939 is a safe and effective option for pain relief. To confirm the direct interaction of exendin-939 with TRPV1, we used multiple approaches, including stable TRPV1 expressing cell lines, pull-down assays, and immunofluorescence imaging with FITC labeled exendin-939. Finally, using patch clamp single-channel recordings, we confirmed that exendin-939 binds to the extracellular side of TRPV1 as a non-competitive inhibitor for capsaicin. This unique mechanism allows it to inhibit TRPV1 activation effectively without competing with capsaicin for binding. We identified a specific fragment of exendin 939 called exendin 2029, yes, which effectively inhibited TRPV1 without affecting blood glucose levels compared to, as you can see, we used exendin 4, GLP-1 receptor agonist as a positive control. This fragment selectively binds to TRPV1, providing pain relief while avoiding interaction with GLP-1 receptor. To conclude, GLP-1 derived peptides, particularly exendin 939 and 2029, demonstrate a novel mechanism for pain relief by directly and selectively inhibiting TRPV1, and their effectiveness is evident across various pain models, including inflammatory and neuropathic pain, making them broadly applicable. Importantly, these peptides avoid the adverse effects associated with the traditional TRPV1 small molecule-based inhibitors, such as hyperthermia. Exendin 939 and 2029 not only provides potent analgesia, but does so safely, offering a breakthrough in non-opioid pain management. I'd like to thank our professors and lab members for their invaluable contribution. Thank you. Thank you very much.
Thanks. Any questions from the judges?
I have a quick question. Yunjing, this was really interesting, and there aren't many great inhibitors of TRPV1 actually, so it's interesting. So what do you think the mechanism is of inhibition? And so this is a peptide, right?
Yes, this is peptide.
Yeah. So how do you think the peptide is working to block?
We think that we first in the preliminary data, we used GLP-1 itself. We think that during the digestion, it's degraded by DPP enzyme. But then those metabolites go through the bloodstream and ultimately go to the DRG, or because we studied on peripheral nerve system, right? So the metabolites of GLP-1 go through the bloodstream, and then it binds to the TRPV1 directly, and we think it inhibits its activations, TRPV1 activation. We think that that's the mechanism for this inhibitory effect.
Okay, but how is it inhibiting the receptor?
We think that it directly binds to the TRPV1. We actually found from the single channel recording that it binds to the extracellular side of the TRPV1. So when it binds to the TRPV1, we think that the ion channel TRPV1 actually closed.
You think it is blocking the pore?
I think the structure changes when it binds to the TRPV1. So it closed. So the channel itself is closed to not not passing the ions through the into the cell, I think.
So it stabilizes the closed state.
Oh yeah, we think
Okay.
so, because
okay.
Yeah.
Thank you.
Thank you very much.
Okay, if there are no more questions, we'll go on to our final speaker in the pharmacology group. This will be Alexa Tolliver, who will be talking about cannabinoids, another talk on cannabinoids. So Alexa, please go ahead when you're ready.
Can you guys see my screen?
Yes.
Okay.
Perfect.
So, hi, my name is Alexa Tolliver, and I'm a postbaccalaureate researcher at Washington University in the lab of Dr. Robert Giraud. And my research is on the expression landscape of cannabinoid type 1 receptors in human sensory ganglia, implications for peripherally restricted analgesics.
Cannabinoid type 1 or CB1 receptors are the most abundant receptor in the endocannabinoid system. Historical, preclinical, and anecdotal evidence all suggest that drugs that target CB1 receptors are effective at reducing pain. So these receptors are located in both the central and peripheral nervous system. However, activation of the receptors in the central nervous system causes the psychoactivity that is associated with cannabis use. So we hope that by targeting CB1 receptors in the peripheral nervous system, we can reduce pain without unwanted psychoactive effects. To understand this, we're looking at behavioral models of migraine, as well as mapping the expression of CB1 receptors in sensory ganglia to understand the implications. So my lab has previously looked at a highly specific peripherally restricted CB1 targeting drug in the mouse model of migraine. So we collaborated with others at WashU to create BIP36, which, like I said, is highly specific and peripherally restricted. And the mouse model that they tested this in is the nitroglycerin model. So mice received injections of nitroglycerin models every other day for 9 days. And then on the 10th day, we administered BIP36 to see how it changed the mice's pain. And to test the sensitivity, we utilized a bond frame method where different filaments with different forces are poked in the center of the forehead of the mice. And if you look at this chart, the 50% withdrawal threshold, the lower the number means that the mice are having more pain-like behaviors. So the mice that receive nitroglycerin at baseline are highly sensitive. However, after administration of BIP36, you see that sensitivity is reduced. And so migraine occurs due to activation of neurons in the trigeminal ganglia. So we wanted to look at CB1 receptor expression in sensory ganglia. We looked at both dorsal root ganglia as well as trigeminal ganglia because they're both sensory ganglia located in the peripheral nervous system that transmits information from the periphery to the central nervous system. So DRG has been implicated in inflammatory and neuropathic pain, and TG has been implicated in migraine pain. So we first started out by looking at CB1 receptor expression in mouse TG, and we looked at CB1 receptor co-expression with SCN10A, and SCN10A encodes for our pain-sensing neurons, our nociceptors, and we see that there is co-expression between the two. We also looked at a subset of these nociceptors encoded by the gene TRPV1, which labels our thermal nociceptors, and again, we see that CB1 receptors are co-expressed as well. We also looked at CB1 receptor co-expression with proprioceptive neurons encoded by the gene PVALB, and again, we see that the two are co-expressed. And then finally, we wanted to look at CB1 receptor co-expression with calc-A neurons. And this is of particular importance to migraine because calc-A neurons express calcitonin gene-related peptide, or CGRP, and release of CGRP is known to cause migraine. We then looked at CB1 receptor expression in mouse DRG, and we see that the receptors are co-expressed in the same sensory neuronal populations as in mouse TG, including SCN10A and TRPV1, as well as PVALB. And then we wanted to see if this data could be applied translationally, and my lab does have access to human DRG tissue. So we looked at CB1 receptor expression in the same neuronal populations in human DRG. So we looked at the co-expression between the CB1 receptors and the nociceptors, and that co-expression occurs in about 2.8% of the total neuronal population. And then co-expression with the thermal nociceptors occurs in about 6.4% of the total neuronal population. However, when we look at the co-expression of all three of these genes, that occurs in about 16.7% of the total neuronal population. We then looked at CB1 receptor co-expression in the proprioceptive population encoded by PVALB, and we see that that co-expression only occurs in about 4.1% of the total neuronal population. So in conclusion, we see that VIP36 is effective at reducing pain in the nitroglycerin model of migraine. And then when we look at the sensory ganglia in mouse TG and DRG, we see that CB1 receptors are broadly expressed in sensory neurons. And then specifically with that mouse trigeminal ganglia, CB1 receptors are also expressed in the calc-A neurons that are important in migraine pain. This work is applied translationally to human DRG, and CB1 receptors are expressed in the same sensory neuronal populations. And we also hope to procure human trigeminal ganglia to map the expression of CB1 receptors in human TG as well. Our next steps also include quantifying the mouse trigeminal ganglia, as well as doing some calcium imaging in mouse and human DRG and TG to understand how VIP36 and other peripherally restricted pain medicines modulate neuronal activity. I would like to thank my lab as well as the NeuroPrep program and the families of the donors. Thank you.
Great, thanks, Alexa.